then we have a week from today, we have our second exam. And we know what it's going to be about. We know what the format's going to be like, the expectations are going to be like. And we're going to start talking about Chapter 19 today. And I just looked at the PowerPoint that the publisher provides, and about half of the images for Chapter 19, this chapter, are ones we've already talked about. Because this is where the author talks about oncogenic retroviruses. Finally get around to really detailing them. And they talk about RAS, and where would you find the mutations in RAS, right? So don't let that make you think, oh, I've got that chapter. Instead, that's a chapter that I mentioned before. That's a chapter where there's probably going to be a stronger kind of expectation that even if we didn't talk about it, you would be able to learn some stuff in that chapter because we have a good foundation going into it. So that's Chapter 19. We're dealing with that. And then you're working on your term paper some, and I'm getting questions for that and stuff like that. And then we have the stem cell assignment video that's due today. I've gotten almost all those submitted already. Then we have our exam. Okay, so then these research internships applications are happening, and it's a good thing. And Juliet was almost on the news. There's a student down at MHR that's going to be on the news. So that's great. All right, this is what Chapter 19 looks like for the topics. So there's the development and causes of cancer, and that's what you would think we would learn about in this course. So you learn terms like carcinoma, sarcoma, lymphoma. So there's some of that, and I won't really talk very much about that because remember I'm a molecular biologist and I don't really care about stuff above that nuts and bolts level. But there's some of that, and we've talked about some of that a little bit as we've gone on, but there's that. Then there's tumor viruses, and that's why the book says, let's talk about oncogenic retroviruses. And we remember that retroviruses are just out in nature, and they cause infection and disease, and randomly, as a mutation, some retroviruses over the years picked up a form of a host gene that was effective, and that specific retrovirus became what we call an oncogenic retrovirus, where immediately upon infection of the next cell or the next organism, it would cause cancer. None of those are known in humans. They were found, Yet. Whoa, set me back. Okay, so it turns out we're pretty solid on that, but we want to be very solid, and we'll go back because there's another oncogenic retrovirus we haven't talked about yet that we want to talk about, and I've already said we want to talk about that one. So we've talked about MYC and RAS and so forth, and we'll pick up a couple more of those and more oncogenes, and the book also has a list of tumor viruses that can contribute to cancer. So that, well, let's see, we need to draw a very strong distinction. There are these classic oncogenic retroviruses that are mutant viruses. There's a whole nother story about a variety of viruses that can contribute to the eventual cancer. And so then it wants to look at oncogenes, tumor suppressors, right? So it's kind of pulling those together. And then it talks about molecular approaches to cancer. We've already seen tamoxifen. That's an example of a molecular approach to cancer. We want to add another really good example that our book just has in a table like that to that story. So all fits together really well. Let me pause and see if people have questions about anything at all. Because we're also reviewing, yes, Emma.
I have two, I guess. My first one is
Oh, that's the best way to start anything. There's a friend of mine that I had years ago, Fred Ponticelli, and Fred would always talk that way. He would always say, I have two things I want to talk about, because that immediately blocks you from saying anything. So yes, Emma, it's a very, sorry, I have two things I want to mention, Emma, and that's one of them, right? So I outclassed you. You have two, I have two, right? But that's a very powerful communication strategy. Like if you ever have children, you don't know how many times I talk to my kids. I want to talk to you guys about two things before you go to bed, and they're like, oh no, two things. Okay, yes, Emma, one is?
so when the book talks about like the Yamasaka factors that mentions like the four, but it also talks about Manog, like is that a part of the factors or is that something apart from that?
So the way for us to think about the Yamanaka factors are that they're a small number. Only a small, misexpressing a small number of genes can turn any cell into a induced pluripotent stem cell. How is that possible? Well, for us, it's not surprising. All of them, 4, are transcription factors. They can have a bunch of target genes. So just a small number could switch a lot of stuff. Then we think about, well, what would it take to turn a differentiated muscle cell into an induced pluripotent stem cell? And we think this. We think, in theory, it could be done because it has genomic equivalence. So our muscle cells have the insulin gene and the hemoglobin gene. They're just not using it. So we'd think that first. We'd think that's a good point. You should be able to do it. But why isn't it a stem cell? And the main answer for differentiated cells is they've added DNA methylation to lots of target genes, and they've left the cell cycle. So when we look at the 4 Yamanaka factors, they're transcription factors. They're going to turn on target genes. They're not, they're not really the thing. They're going to turn on the thing. And so the thing would be, we got to get back in the cell cycle. And that one of the 4 is MYC. And we're just like, oh, wow. But that just underscores how astonishing MYC is. You've left the cell cycle in your brain. You're in that G0. You're not going to go into the cell cycle again unless you were forced to express MYC. And it's not a signaling factor. It's like the force expression inside the cell. That's not a natural thing, to have. But, it makes you think, oh, wow, that MYC, we've learned it's a dangerous oncogene. And that's kind of underscoring that. Then the other 3, we don't have to worry about those too much. Our book talks about them a little too much. For us, we'd want to emphasize that the other 3 are involved in a bunch of things, but including activating expression of genes that would be demethylases, that would start removing methyl groups, because we know that's something you gotta do that, right, to get to reactivate that. So that's a key feature for us. And then that nanog, there's a complicated interplay because the cell has feedback mechanisms to try to not have a differentiated cell suddenly become cancerous like that, start to grow out of control. And so it's not as easy as just like, oh, and then activate these 4 transcription factors and you're rolling. There's a complicated interplay between those and additional transcription factors like nanog. So pretty quickly you get down into details that you could understand based on the stuff we're learning in our course, but they are like getting into the weeds, like that, where it's advanced stuff and it would start to maybe cause you to start getting confused, like the question. That's your first one.
Yeah. Second, or you want second one?
Yeah, you are.
So the book also talks about like the pro-apoptotic factors, but then it goes into like the subcategory of the effector ones versus regulatory ones. Do you want us to know that difference first?
No, because I've always tried to emphasize that effector term is just kind of a loose term that we just want to be gentle on and not really memorize and sort of rigorously insist, oh yeah, that's it. It just helps a person appreciate that, like for programmed cell death, we've got a complicated upstream regulatory pathway with all of the different BCL2 factors playing a role, pro-apoptotic, anti-apoptotic, a lot of feedback loops. But we're going to get to action, and caspases are the action. And want to keep it very separate, but over at the cell cycle chapter story, we have all kinds of upstream regulatory stories, retinoblastoma and stuff like that. But we eventually get to CDKs, and they are action, both at start and also the G2 to M transition. So over at the G2 to M transition, we have cyclin B and CDK1. Are the only letters and numbers we named for those. And when CDK1 is active, you're in mitosis because it's just doing phosphorylating the targets along with Aurora and Polo to just do all of that stuff. Just yep. So yes, so yeah, so you could say, when you start saying, like if you really start digging in with like this is an oncogene and that's a tumor suppressor, those terms also, become a little bit fuzzy when you really push them like that. And that's fine, but for our course, it's a really important principle, and I'll show you that there's just one place in our course. Maybe I'll scroll forward to this slide. It's way toward the end of this chapter, but the book shows this for this purpose here. Let me just see if I can. It's pretty obvious slide. Oh yeah, we have some learning to do before we have our exam. Oh yeah, here it comes. There it is. So this is this familiar pathway where we saw that the PI3 kinase could get activated by a receptor tyrosine kinase back in chapter 16, and the PI3 kinase would phosphorylate PIP2 to make PIP3, and that would activate AKT, right? Like that. So in general, AKT, for most kinds, it stimulates cell proliferation and survival, okay? So the idea is the PI3 kinase could be considered an oncogene, because if it got mutated, you would overstimulate that. PI3 kinase plays a role similar to the role of RAS in the other pathway, right? That if you get mutated in RAS and it gets to be doing its job too much, so you could, in principle, do that, see that, right? So what the book brings up here is, I think, what you're asking about a little bit, and that is, we learned that for every kinase, there's a phosphatase that does the opposite. It removes the phosphate. So if this is an oncogene, wouldn't the corresponding phosphatase be a tumor suppressor? And the corresponding phosphatase in this case is called PTEN. And, based on our understanding, yeah, it's a tumor suppressor. And that's seen. If P10 gets mutated, you can have too long of an activation of PI3 kinase, too much stimulation. It's the same story as the RAS story. You just get it going too much, too long. And you can have that by having PI3 kinase, or you could have a mutation in the P10, because it would not remove the phosphates, right? Like that, and it would stay active too long. So that's a thing, right, where you start really getting into it and being like, is that a tumor suppressor? And we would say, yeah, definitely, definitely. But it also is like, just like, oh my gosh, there's a lot of oncogenes, then they have opposing, tumor suppressors, And that's what I mean by fuzzy. It's a good definition, it's real and true, but most people wouldn't jump up and say, oh yeah, P10, that's a tumor suppressor, But yet, yeah, it fits. And so our book is bringing that up for this purpose too, is to kind of take a more advanced appreciation of what the meaning is for an oncogene and a super suppressor, because there are lots of proteins and genes, corresponding genes, that are literally either potentially oncogenes and tumor suppressors. We've seen that some oncogenes and some tumor suppressors are very significant in specific cancers. And as scientists learn more details about those cancers and the remaining cancers like that, so we talked about ER-positive breast cancer, and they found other ways to treat breast cancer with repressing other growth factor receptors. And now there's a small population of people who get breast cancer that is referred to as triple negative. Negative for ER, negative for the next one, negative for the next one. So what's their cancer? And so doctors and scientists are getting down to some cancers that are, problematic, partly because there is a sort of a detail where you're like, that's, it's a tumor suppressor, and that person has a mutation in it. We have to try to find a way to treat that one now. It's like that, because there are just so many different ways that you can either be enhancing growth, suppressing growth, enhancing apoptosis, suppressing apoptosis, enhancing DNA repair, like that. So yeah, so that's that. So we don't need to dive into that like in great detail, but certainly the terms proto-oncogene means an unmutated natural one. And I'll just draw this out because we kind of glassed over that because we were, foreshadowing. But when scientists talk about wild type MYC, sometimes you see they still would like to do this, and they put a C there, and it stands for cellular MYC. And that just means wild type MYC, the normal MYC. And what they're actually going back to is they're actually going back to the terminology of the oncogenic retrovirus scientists from the 1970s. And so you don't see this very much anymore, but it just pops up. And in that, those scientists would want to distinguish. We got the oncogenic MIC, and that we want to call VMIC, because we found it in a virus. So that's that terminology there. And and so nowadays, instead of referring to it as CMIC, more commonly, and Emma's made this to use this term, people call that a proto-oncogene, and that means it's the normal wild-type gene, and if it got mutated, it could contribute to cancer. So we're walking around, all of us, hopefully with a normal version of MIC, a proto-oncogene that could get mutated and cause trouble. Yep. Other questions people have? Yes?
What's the difference between a proto-oncogene and just an oncogene?
Yep. So the people use that term to just distinguish between these characteristics, right? So if a person said an oncogene, they're not really saying whether it's the normal version that could get mutated or the mutated version already.
Okay, sweet, thank you.
Yeah. So that's the whole proto-oncogene, to really say, we're not talking about the one that's out of control, we're talking about the normal one. And so, yep.
Does that mean that the term proto-oncogene is entirely subsumed within the larger category of oncogenes?
Sure. What did you, yeah, sure, yep, yep. Yep.
So I have a question that kind of builds off of
No,
that.
that's a legit question, yes.
It's about categorizing stuff, because the caspase, I was studying this last night and I kept getting really confused. Because you say that the caspases are a type of a specific, what's the name for the proteases? But
Yeah.
they normally don't have specific places that they cut, but then you say that the caspases specifically cut the scramblases to activate it. So I was wondering, category-wise, is it like the family name is caspases and then a specific type of caspase cuts the scramblase, or?
No, got it. So there are enzymes that are called proteases, and proteases degrade proteins. Or they cut proteins, I shouldn't say degrade, they cut them. So proteins join together, amino acids join together by a peptide bond, covalent bond between them. And all proteases, their function is to cut. Then of all of them, each one has, like all enzymes, it has a specific substrate and a specific catalysis. So there are digestive proteases, that are released into our intestine, and their goal is to just completely degrade the proteins we've eaten into individual amino acids.
So those would be the non-specific ones.
They're specific. They're always specific. They always have a specific substrate. But a lot of those have pretty liberal cutting strategies, like they'll cut after every glycine, right? So those are proteases, and they're digestive proteases. We're trying to break these things down to amino acids. And a lot of times people, students have kind of heard about that idea. We got to degrade proteins. Proteases degrade proteins. A lot of people know that some proteins go into a big organelle called the proteasome, and they get digested, broken down to individual amino acids. So they're a little bit surprised when they learn that there's some proteases that aren't digestive, they're regulatory. And what they do is they just cut a protein in a very specific location. And in the case of caspases, they don't degrade proteins, they just clip them. And those proteins that are the substrates have started out and they are inactive. Right now the scramblase is inactive, and it's actually referred to as a pro-protein. And so, yep.
And are we using proprotein as the same as zymogen as a synonym?
I don't know what what's zymogen mean?
Like a protein that needs to be cleaved in order to become active?
No, because you could take insulin, for example, and insulin, it gets secreted. And so you might know that insulin gets synthesized as a pre-protein because it has a signal sequence that targets the ribosome to the rough ER to complete synthesis of the insulin pre-protein in the rough ER. And that's taught hopefully in intravile. Insulin, mature insulin, actually gets further processed in the rough ER and the Golgi to remove parts of it. So initially, insulin is synthesized as a pre-pro protein. And so, I already forgot what you said zymogen was, but that's fine. It's just this specific idea that Cas bases do not degrade proteins, they just clip them. And there are specific substrates like scramblase and the inhibitor of the DNase, they just clip and it activates it. And before it gets clipped, scramblase is referred to as a pro-protein. What's a zymogen again?
A protein that needs to be cleaved in order to become active, like.
Yeah, so there would be
pepsin.
pre-proteins, those all have to get cleaved before they're active because that's part of their synthesis process. So I would say zymogen, I would say that pro-proteins and pre-proteins are completely subsumed under the term zymogen. And there would be others that would be under the broader term zymogen. Whew, I dodged that. I wish that I'd get fired. I really do. That would just be so funny where you're like, you're out of here. And I was like, what? He's going to go work at McDonald's. Okay, so other questions people have. Yeah, try to get me fired. Yes? Yay!
In what way is it useful to think of cytochrome C as a second messenger in apoptosis pathways?
I don't think anybody would call it that. Yeah, because it's, and that second messenger concept is another example where there's some that are classically defined because of the history of it, where they're like, biochemists were really, didn't have a lot of powerful methods, but they could analyze these small molecules and measure their abundance in a cell, and they would see that cyclic AMP would go up before they could understand anything about the other proteins that would be in that signal transduction pathway. And so, so it's a term, and it's a legit term, but it's not one of these that's just like rigorously defined and used like that. So, so it's one of these things where you'd be like, how would you know? How would you know? How would a student know whether cytochrome C would be a second messenger? It's a small molecule that gets released and floats around in the cytoplasm. That's what happens.
From the mitochondria, correct? From
the mitochondria, yep. Leaky mitochondria, yep. And it's just, convention that people wouldn't just be like, and that becomes a second messenger, yep. Yeah, that's a good question, right? Because how would a student know? And it's there because there's some convention, right? Where they're just like, no, people wouldn't call that a second messenger, even though it fits the idea perfectly, yep. It's not as, a pathway like that, yep. Another thing I would say about that is the term signal transduction pathway, I think always refers to signals that originated at the cell membrane and are being communicated into the cytoplasm. And sometimes apoptosis is triggered not from an external signal, but from an internal signal. And so that cytochrome C would be on a pathway that didn't come from the outside and from what people would say is a signal transduction pathway. So even though it seems a little bit like a signal transduction pathway, it's completely internal to the cell, and those aren't ever referred to as signal transduction pathways. They're just a pathway.
But the cytochrome C activation.
Fire me! And get like a tuition rebate where he got fired and I don't want to pay for the course.
So the cytochrome C activation happens both with external and internal inducement of apoptosis.
Yep.
Okay.
Yeah, it's a big feature. Yeah. I don't know if it's universal or not, if that always happens, but I think people would say there's always mitochondrial involvement, so probably. Yep. Other excellent fire me questions? I'm sweating. Just saying. Yep. Yes, Osira, take a shot.
How did I understand the mitochondria's role in apoptosis? And I was confused in the video as well.
Yeah. So it's and that's because there's a bunch of roles. For us, this term, the mitochondria becomes leaky, is the key. The mitochondria has a phospholipid bilayer around it, a double bilayer, right? So things are not coming out of the mitochondria. And so for us, the key thing is that when apoptosis is going to occur, lots of times it includes those BCL2 family members, some of which are bound to the mitochondrial membrane. It includes those being activated if they're proapoptotic, and they cause the membrane to become leaky, and things leak out, and the one thing we learned is cytochrome C. And that's really it. It's complicated because it becomes a story of those BCL2 gene family members, and some are proapoptotic and some are anti-apoptotic. And so we kind of gloss over that because it would just be confusing to, we're also not learning the names of them because it's just a complicated interplay, a balance between them. And so again, for us, the mitochondria becomes leaky, and the stuff inside the mitochondria leaks out, and cytochrome C is one of the things that leaks out.
Can we think of it as the acid? Almost.
Nope.
Because that's kind of how I was thinking, like, it's like it releases this acid, which then kills this.
No.
No, okay.
Nope. Yep. Other questions people have? Yes, Usira.
So there's cytochrome C that, is it like an internal signal that tells the cell to die?
Mm-hmm. It's on a pathway toward activating caspases,
Okay.
right? Remember that I showed this picture with color balls about the pathways of programmed cell death, and one of the proteins is caspase and another protein is BCL2. And I showed it because it showed 3 different species. It showed this very simple nematode worm, a fruit fly, and a mammal. And I wanted to show that to emphasize the orthologs found in all animals, BCL2 and caspase. And definitely caspase is found as an ortholog in every animal, but we also saw the nematode had a relatively simple regulatory pathway, and then the fruit fly more complicated, and the mammals more complicated, because that's what happens when you have evolution occur to make more complicated organisms, is you tend to get more complicated regulatory pathways because they have more circumstances. Remember that we have blood cells that are supposed to die if they don't get activated. So that makes our story of apoptosis more complicated than the nematode story of apoptosis. And so animals need that, but some have a more simple need for it, not so many examples and diverse examples, so they just have a simpler pathway. Yeah, but that's what we wanted to get from that. Yep. Yep.
And with the arrows in that figure that
Yep.
you showed, was that to show that CED9 and BCL2 are like orthologs, and then CED3 and caspase9 are orthologs?
Yes,
Okay.
but remember that a student asked a question about the names, and we don't need to learn CED3 or CED9.
So what should we know?
BCL2 and caspase.
Even in the nematodes, because
Mhm.
it said, okay,
Yeah,
so
yeah.
they're just as.
Yep. Yep. More questions? Yep. Yes.
Is there a story on why the stem cells in mammals differ from other organisms? Wasn't it?
Yep. Only mammals have embryonic stem cells. Yep. Yeah. And that's dealt with a lot in the course I teach in the fall, developmental biology. But it goes like this: mammals have internal fertilization. So mammalian eggs are tiny. They don't have any yolk nutrients in them. You know, a chicken egg has a big yellow yolk. That's the egg cell in a chicken. And it's packed with nutrients, and that's true for all non-mammalian organisms. They have big eggs, relatively big eggs, because the mom adds nutrients. And in all those non-mammalian species, the mom also actually adds specific factors, proteins that are going to play a role in early embryonic development. And because of that, there's no way to divide the fertilized egg up into 2 and make the 2 cells equal. Since mammals don't have big eggs, they have tiny eggs, they don't have those nutrients, they also don't have those factors from mom. And so the 2 cells are equal, the 4 cells are equal. And so that's the reason why only mammals have embryonic stem cells. That's another way to say that is mammals are the only animal on Earth that could possibly have identical twins. in nature. You know, maybe somebody could produce one in a lab because it's cloning. Identical twins are making clones, but not in nature.
What
It's
about
kind of
the eggs with the 2 yolks? Have you ever cut?
So those are fraternal twins.
Oh.
Yeah, so remember, fraternal twins are 2 eggs and 2 sperm. Identical twins are 1 egg, 1 sperm. Identical have exactly the same genes. Fraternal twins are like siblings, no different than a sibling. Yep. And so you can have 2 eggs and 1 yolk. Yep. And that's what's that called? Christmas Eve omelet. Yeah.
Is it even possible for 2 chicks to develop inside that same egg with the 2 yolks? Is that
That's a good question. I don't know. You see those double yolk eggs, but I don't know if they've been like successfully fertilized and would develop and come hatching out. Yeah, I don't know about that. Yep, that's a good question. Yes, but again, that's kind of emphasizing the fall course. Other questions people have?
Do you care about the difference between totipotent, pluripotent, and multipotent stem cells as different terms?
So I haven't used those terms because they've been sort of damaged in their use because of induced pluripotent stem cells. And so people use them, but we don't need to deal with that. Yep, we can talk about it, outside of class and talk, because it's kind of a bit of a mix of history and use like that, which is, it's unfortunate when you're teaching or learning a topic, but yeah. Yeah, they're called induced pluripotent stem cells, yep. Other questions people have about anything at all? All right, so we can keep asking them, right? Because you're thinking about the exam and getting ready for that. That's great, okay? So back to the beginning. Awesome, thank you. And I joke around sometimes, but questions are great. They're always just totally worthwhile to bring on, whatever they are. Okay, so then these different areas that we're going to roll through. And so here's just the first example, and I realize it's really too small to see, but they're talking about a cancer developing, a tumor developing in cells, and looking at under a microscope. So kind of a clinical, doctor-like story about cancer. Here's something you don't need to know about. These are some carcinogens, so molecules that can cause cancer. That's not our focus. You can certainly ask questions about it, and if we talk about it, it might become a 2-point question. But I wouldn't put that on our exam. And I show that because as having taken a bunch of chemistry, students would key in on that and be like, okay, I can memorize that chemical structure, right? So then it goes into oncogenic retroviruses. And we've seen this before, right? It's a normal wild-type retrovirus that has retroviral genes that it needs to make. For example, the polymerase, the reverse transcriptase that copies the RNA into DNA. And the problem with an oncogenic retrovirus is that the virus inserted into the host chromosome randomly and happened to insert next to an oncogene, like RAS, and it accidentally took it with it. So in that one infection cycle, in that one cell, it became an oncogenic retrovirus. It's a mutation. And the problem with it is it picked up a host gene. And there are about 20 of those or so that were known and that had been found in nature over time, and they were not in humans. They were in cats and chickens and mice and rats and monkeys and so forth. But that gave them this head start, a foot in the door in the 1970s. And our author, as we talked about, going from Wisconsin to Boston, played a big role in it. And here, he doesn't name himself, but he describes it. He describes his experience when he was your age, because he shows this is the event when the retrovirus became oncogenic. So, it infects this mouse, and then it actually picks up, in this case, one of the other ones that we're not going to worry about, Abelson, and it's a murine leukemia virus that it's going to cause, and it picked it up. So that's the event. Right? These were just normal retroviruses in nature causing infections and disease, and then it picked up a gene, right? So that's that gene. And then he talks about it, there's the oncogene, right? He writes that explosion around it there. These are oncogenes they're picking up, right? You wouldn't pick up a tumor suppressor gene. That's not going to be problematic. If you add that to a cell, you're just going to get more tumor repressor, even if the tumor repressor was mutated. Your tumor suppressor is still there, So only oncogenes could be trouble, like that. Then he shows in RAS, the oncogene in the virus, it is mutated in the coding region, specifically in the catalytic domain, the active site of the enzyme that's going to inactivate the RAS by changing the GTP to GDP. So he's just talking about that, and you need to read that carefully. And you know, it's spiral learning. You're like, oh, wow, I didn't quite get this until now. I'm really getting it better. Yes.
Zooming in on this, Dave. This story is that the virus has picked up a mutated RAS gene.
That's a good question. So the deal is that we know the RAS has these specific mutations in it. That's the problem. It's not that it just picked up a gene, but it's got a mutated gene. And so scientists would say what really is likely to have happened is it just picked up a wild type RAS, and it wasn't, in this case, it wasn't initially problematic. But viruses don't do very much DNA repair. They just get copied really fast, and if you make a bunch of viruses and some of them are just defective because of DNA errors, you know, who cares? So viruses that picked up a RAS would mutate it really frequently, randomly, and then that's the one that would really become a problem. So that's a good point, that virus probably might not, no one knows, but it might not have become initially problematic. It was just carrying this extra gene, the RAS gene, but it would just get randomly mutated, and the virus wouldn't care if it was mutated. It doesn't help or hinder the virus at all. And if it got the mutation in the active site, well, then it would be contributing to that out of control thing, even if the infected cell had its normal RAS genes and RAS proteins. You're making this extra defective RAS, and that's going to cause trouble. Yep. All right. So here's just a graph that you might study for a long time, but these scientists were working a long time ago, and they didn't understand the genes very well. They didn't have the methods to read the DNA sequence. And so here they're doing is they're actually just taking different animals' DNA in a test tube, and they're heating it so the DNA unzips, and they're cooling it, and they're asking if these oncogenic retroviruses will stick to the normal animal's DNA. And they would. It would stick. And that's the evidence they got that it was a host gene. And then they did the same experiment with different species. And they found that the oncogenic retrovirus, say from the chicken, would also stick to the quail DNA and the duck DNA. And that was the first hint that these were orthologs. And that term wasn't around there, and that concept was not really around there. But it showed that if you found an oncogenic retrovirus from a chicken, you could take that, and that DNA would stick to a normal chicken's DNA. It was a normal, there was something in the retrovirus from a normal chicken. But it would also stick to quail and duck and monkeys and other animals. Remarkably, you know? And so our author did that work in Wisconsin, and then the author is like, okay, so these are host genes. So I'm moving to Boston, and I wonder about just taking just a mouse that's healthy, and I wonder if I could just grind up some DNA from a mouse that has a tumor. So take out the mouse's tumor, not caused by a retrovirus, just a mouse that's got a tumor. Could I grind that up and put that into a mouse and have that cause cancer? So leave the retrovirus behind, but use the same logic. Could we have a host gene from a tumor get put into a healthy mouse and immediately cause cancer? And so they show that here. Nope, not quite yet. Here. So here they're just taking pieces of a tumor and cutting them up and then putting them into healthy cells and finding cancer. And he did that, and he found that the DNA that was causing the problem in his case was RAS. So he studied RAS in Wisconsin in a retrovirus, and then he moved to Boston and he found RAS in tumors that was defective. So it made that, and other scientists made that bridge, that it wasn't just a story of retroviruses. These were just the regular tumors that people and animals have in them have defective genes that are causing cancer. And they found over and over again, it's the same, gene, RAS in that retrovirus or RAS in that tumor. Yep. Yep, okay. This picture I jumped over is just talking about, the idea of the virus, again, back to Wisconsin, and the virus carrying a defective host gene causing trouble. Yep. And then the book talks about that, and then the book also talks about the translocation. So why is it defective? And so the book talked about how RAS is defective, it's mutations in the coding region. Now it's talking about some tumors that are defective because of a translocation. So it's MYC. And other scientists had found MYC in retroviral oncogenes, and then they found it in a tumor. And when they found it was a tumor, it was like translocated and things like that. So that's a really old school thing too. And remember that scientists had already seen that under a microscope, because you can do a karyotype and see chromosomes under a microscope, and you can look at them in people who have cancer, and in some cancers, like Burkitt's lymphoma, you see a common chromosomal translocation. So that was another big connection way back. And he's showing these old school connections that we understand. He's just putting them in. And for us, it's a review and a little bit of an add-on like that. And he gives additional examples because some of these other oncogenes are super famous. So here's that Abelson, and it's going through a translocation to produce, it's a lymphoma. And that Abelson, Abel, we don't worry about that. I said, we're going to learn some names of these. And there's one more we're going to learn, and it's not Abelson, okay? So, look at that. It's a chromosomal translocation. When we think chromosomal translocations, we have great examples. The woman with skin cancer, the people who have Burkitt's lymphoma, the people who have non-Hodgkin's lymphoma, right? So I could say, describe 3 examples of chromosomal translocation. And if you used Abelson, fine, right? I'd be like, wow. But I wouldn't directly say, draw this picture, because we have ones that and you're like, I understand what's going on here because I understand the MYC story and the BCL2 story and the PDGF story, right? Like that. So think about it that way. And they're talking, there's the chromosomal translocation between the regulatory region of BCR and the oncogene Abelson. And it was seen under a microscope a long time ago in Philadelphia. So they call it the Philadelphia chromosome. Don't need to know that, but it just shows you how old school those karyotypes are. They're like, it's that chromosomal translocation that those dudes found in that tumor in Philadelphia. That's that cancer. You have that cancer. Yep, okay, great. And just, more, they just grind into some more detail that, that looks like a lot, and it is, but nothing, we need to get out of it. So then, here's the next thing, is we want to talk about now, let's go with this first. So we've already seen this too. This is where I said our book shows the June and FOS. So we're used to the idea of a receptor tyrosine kinase, like PDGF receptor, activating RAS, and then the MAP kinase pathway, RAF, MAC, ERK, ELK, and then ELK now with SRF activating, we like to think of it activating MYC. But in other settings, other contexts, it activates FOS and June, and those are also oncogenes. So we have a little detail there, that's good. So we learned that the June FOS heterodimer could bind, and we could draw a picture on our exam of a cyclin gene, kind of a general cyclogene, that would have different enhancers. An enhancer to bind beta-catenin, an enhancer to bind MYC, an enhancer to bind June FOS, right? So our book is showing us that. That's where this comes up. And it does it because June and FOS are also famous oncogenes. Would it help for us to really learn a great deal about them? No. But here's where we're going to just plug this in. So here's where the book shows, beta-catenin. Look at beta-catenin. Now, I've showed this before. Beta-catenin activating MYC, activating cyclin D. Right? Beta-catenin and Wnt, they play a big role in all kinds of different stuff. It includes stimulating cell proliferation. So they're gonna do that, yep.
Beta-catenin is the one that has orthologs in like all
Yep.
mammals.
The
or
whole
only-
Wnt signaling pathway, so Wnt and its receptor and the regulation, the signal transduction pathway, and then beta-catenin. Yeah, it's just crazy.
Is it all mammals or
No,
all?
every possible animal you can find. Yep, yep. Yeah, jellyfish have orthologs of that signaling pathway. They're really distantly related to us. And it's not because they have cancer, it's because that pathway plays a big, big role in lots of different pathways. Yep, that's true, okay? So then, let's just move to this. We're doing great. But let's go to this final oncogene that we're gonna learn, final retroviral oncogene that we'll learn about. And you see here, this pathway, it says there is a growth factor receptor. It's a receptor tyrosine kinase. Okay, so we know receptor tyrosine kinases because we know about PGF receptor, insulin receptor, KIT, and KIT receptor, which has a name. And then here's our last one. And you don't have to learn its regular, normal proto-oncogene name, but I'll just say it. It's epidermal growth factor. It normally responds to epidermal growth factor, so it's EGF, the EGF receptor. So a lot like PDGF, but we're going to learn its oncogene, its retroviral oncogene name, and I'll write it out in bigger letters. It's called ERB-B. So it's one of those, and I'm just going to scroll to the place, and we're going to talk about that. Okay? Okay? So, back in our course, we saw this, retroviral oncogenes that we'll especially learn a lot about. RAS, KIT, the KIT receptor, mixed transcription factor, a little bit of Jun and Foss transcription factors. Way down here is SARC. SARC comes up kind of a lot in this chapter. We are just not emphasizing SARC. We talked about it a little bit. It's not a big story. It's this adapter. It's the SRC homology domain between the receptor tyrosine kinase and things like RAS. But here's one, ERB, and I wrote it out here, also called the epidermal growth factor receptor. And it's another receptor tyrosine kinase. So ERB-B. And it was found as a retroviral oncogene. And I'll just draw out what happens with ERB-B when it's mutated. Because it's a little different thing. It's a very familiar kind of situation where here is the gene, there's the and the there's the coding region, and it's going to code for a receptor tyrosine kinase that we're going to call ERB-B. And it's one of these oncogenic retrovirus host genes that is problematic. And this is what the mutation looks like when it joins or gets the retrovirus, or if it's mutated in cancer. And that is, in this case, the breakpoint's right here in the coding region. We're used to a situation like MYC, where we have MYC, and then we have the retrovirus regulatory region, making too much MYC. Here, what's going to happen is we're going to make not a hybrid protein, like we learned about how we could make a FOXO-GFP hybrid protein. We're going to make a shorter protein. So it's just going to be missing part of the normal protein. And that's going to make it cause cancer, okay? And we can draw a picture of what part is missing. So there is the cell membrane. And here's what the defective ERB-B protein looks like. It doesn't have much of that external thing. It can't bind EGF. So this is the mutated version. And that's going to cause cancer. And it's normally a dimer, so people would draw two of them. And it's a receptor tyrosine kinase. It's one of those, right? We have 58 of them, which is another one of those. It's normally called EGF, but this is the mutated version. and it's just missing a part of the protein. I guess I should sketch that in. You know, there's this part of the protein that just is not there. It lost that part of the coding region, and it would make the extracellular domain. And that's usually really important for it to be activated. But unfortunately, just because of the structure of this one, when it's shortened like that, and when people talk about a shortened protein, they usually refer to it as a truncated. That's just a normal English word, not a science term. But it's a truncated protein. It makes a truncated, shortened protein. And when it makes that mutant form, it is constantly active. So it is going to constantly activate what? RAS. So that's what it's going to activate. It's going to activate that familiar RAS, RAF, MEK, ERK, ELK, MYC pathway all the time, because it's just defective. And it's just defective, and it's weird because it's missing part of the protein. How could that make it defective? It's just made it defective. Is that seen on all the other ones? Nope. It's something about this one that makes it kind of vulnerable, like kind of an Achilles heel. Yep.
So it's missing the pliers part of it,
Yep.
basically, and that was what would cause the cross, so it's just constantly cross-correlating.
And I think the idea is that normally we need to have these two external regions come together when they bind the ligand, and that brings the tails together, and then they autophosphate. Here I suspect that the truncated form just makes a defective shape, and it sticks. You know, it's like gluey kind of glob. It just globs together. I'm not sure about that, but it's probably something like that, that it's just defective like that, okay? So I have some slides from a thing. So this just shows this, you know, just trying to show that. So it's showing here, and it's obviously not from our book, the normal receptor has that domain, it binds to the ligand, and it activates it. Here's that truncated version, and it's constantly active, and that's what we call ERB.
So you're just gonna be making MEK, tons and tons of MEK.
It's making a lot. Yep, yeah. It's activating the same pathway as PDGF in other cells. Yep. Okay. All right. So that was, people were faced with that. There's a cancer in humans that has that problem. So they originally found it as a retroviral oncogene, and then they found tumors in people that had that same type of truncation of the ERB gene. And so they're like, okay, that if we could stop that protein, we could stop that growth, we could stop that cancer. So because it still has a little bit of the extracellular part, some scientists at a drug company made antibodies that would stick to that protein, the defective version. And they found an antibody, and antibodies are just proteins that will stick to specific molecules. And they found an antibody that would stick to the defective one and turn it off. You know, just got lucky. They went fishing with antibodies, and they found an antibody that would stick to that defective ERB protein in a cancer and turn it off. It was still there, they would just turn it off, you know? I don't know how, but one way is if it's stuck before the two parts glob together, it would turn it off. So it's something like that. It just blocks it from getting turned on in that defective way. And the antibody is sold as a cancer drug. So we learned tamoxifen in breast cancer. Remember what that is. This is a different kind of treatment because it's an antibody. And this drug has a name. And so, reminder of the tamoxifen story and how that's only used for patients that have ER-positive estrogen receptor, cancer. Same with this drug. This antibody is only used on people who have mutation in ERB. They couldn't use it on other kind of cancer because that's how it works, right? It has a specific molecular mechanism action. Our book shows this table in chapter 19, and it's showing this idea. It says represented oncogene targeted therapies, and here's ours, and I'll have it in bigger letters, and it says it's targeting ERB, and the drug is, there are different names for the same drug, but the name we're going to learn is called cetuximab. And the last part is for AB for antibody. It's a monoclonal antibody, and it is used only in very specific cancers, like that, okay? And so you don't need to pay attention to this slide very much. This is just showing a summary of this, and it's showing that here's this normal, or here's the ERB receptor, and it can activate the RAS-RAF-MERP pathway. There's that SARC homology domain that we don't worry about too much. And I just wanted to show you this because we're familiar with that. And we're like, yeah, that would be a problem if you activated it. It's also activating a JAK-STAT pathway. It's also activating the mTOR pathway. And I show that because even though it's not showing that in the detail here, it simulates cell cycle progression and anti-apoptosis. So this is a dangerous receptor to have fail because it's both saying, go through the cell cycle and don't worry about apoptosis, you're in good shape. So it is a little bit of an Achilles heel with regard to its structure, that you could break it there and get it going out of control, and also Achilles heel, like a weak point, because it activates both those pathways. So it's a dangerous receptor to get in trouble with. And then it shows this word up here, and I'll write it bigger. There's our antibody, and it's a drug that a person would get injections of to treat the cancer. And so here's just the spelling of it. So we learned tamoxifen, and that's a drug that has a lot of different trade names. It's using different names, but here's ours. It's called, and the book has it in that little table. And I don't know, Satuz, I think it's an X, a MAB, and it's an antibody that blocks HERB. Okay? And that's why that receptor tyrosine kinase came up, is an example of using an antibody in therapy. And you can see another company has a different one that's used, so these drug companies are competing. They're like, that's a good idea. We should try to do that too. Here's cetuximab, and this is the reason we're talking about it partly, and it's this highlighted thing in the Wikipedia page. So cetuximab, it's also sold that, is an epidermal growth factor receptor inhibitor medication used to treatment of metastatic colorectal cancer, head and neck cancer. Cetuximab is a chimeric monoclonal antibody. Imagine that you have a friend or family member at some point in your life that says, I'm getting some cancer therapy and they're giving me shots of this drug called cetuximab. And so you're going to look it up and learn, because that's what happens. When you have taken some college classes, you'll have friends and family members for the rest of your life saying, hey, Smarty Pants, I'm in trouble. Okay? So you could look at that. And then look at this, because it's super interesting. So in July of 2009, so 15, 16 years ago, the FDA approved cetuximab for treatment of colon cancer with wild-type RAS. And so that's our key concept here. They said, yeah, you can use your new drug to treat cancer, but only in patients that have a normal RAS. Because remember, RAS is mutated in about a third of all cancers. And remember that RAS is downstream of the receptor. And the FDA is saying, you can use this drug, it will help people, but it would not help a person who also has a mutation in RAS, because that is a problem that's downstream. Logically, it's a real simple concept, but it's really significant to appreciate that, right? That it just wouldn't help if the person also had a mutation in RAS, because the cetuximab would not slow down the RAS, it would just keep rolling. And that happens frequently. The FDA realized it's a good chance those people also have a mutation in RAS, because cancers typically have multiple mutations that really get them rocking. So they said that. You can use it, but only if they have a wild-type RAS. Does that make sense? And then it says this, that you can only use it in RAS, a wild-type RAS, because it had little or no effect on colorectal tumors harboring a RAS mutation. Okay? And then it says this, 2009, right? This was, where does it say it? This was the first genetic test to guide treatment of cancer. In July 2012, so 13 years ago, the FDA approved a PCR companion assay to be used with the cetuximab drug to test for RAS, right? So you got to make a product, right? The company's like, we got a product, go, let's go, let's go. And the FDA says, wait, we're not going to let you use it on anybody because you would just be making money for fun. Because you would not be helping people if they had a RAS mutation. And the company's like, we go back to lab and make some kind of assay to test if the RAS is mutant or not. Be able to sell our cetuximab. So that was not that long ago, right? We've been learning about this kind of talk and this kind of logic, this molecular mechanism of cancer, the whole semester. It's all really fairly recent stuff. Okay? So that's why we come to that. It adds a thing. Okay? Then it also adds this. Oh, and I also have to find this slide, and I hope I didn't severely misplace it. There it is. So this is common treatments for cancer. And you've heard of the first one probably, radiation therapy. I'm going to get some radiation therapy, going to focus on this cancer, try to kill it with radiation therapy. Then there's chemotherapy. That means use some drugs, give the patient some drugs to try to stop the cancer. And we have just really one example of drug therapy, it's tamoxifen, but there are many, many, many. This one people call immunotherapy. We're using an antibody, a product of the immune system, as a drug. It's not a kind of traditional chemical drug like you could synthesize in an organic chemistry lab. It's a protein antibody, and that's called immunotherapy. And then our fourth one, we've already talked about, that's the topic Sam brings to the class, cellular therapy. So that's important for us to appreciate, and there are many examples of the first three, and more all the time. They have really different strategies, right? As scientists get more and more specific in their targeting, they get more and more specific in their strategies. That radiation therapy, that's a lot like surgery. I don't have that up there, but that's another classic one. That's kind of old school. You know, we're just going to go after this, try to cut it out or try to burn it up, you know, kind of like that, right? Then this chemotherapy, that's drugs. And the drugs started out simple. They're just going to be, you know, trying to like block DNA synthesis and things like that. Then they get fancier and fancier, like tamoxifen is a modern fancy one. Then now there's this immunotherapy, and there's a variety of different immunotherapy strategies. Our immune system is very powerful. We're each getting rid of cancer frequently because we're relatively young, you are relatively young, and have strong immune systems and can identify cells and wipe them out. The whole point of that CAR T cell therapy is try to boost that immune response, that natural immune response. So these are more modern treatments. So we put that in there also as an example of this immunotherapy. Doing great. Questions about this? So I'll go back to that, wherever that went. I guess we had this in kind of the wrong place. Yep. I'll just show you this. This is the one place where the book talks about CAR T cell therapy. Remember, on this exam, we're really focusing on the cellular responses in that CAR T cell therapy. So let's see if we can try for a little concept chat. And we only have a few more weeks left in the semester, so see if you can meet somebody and say, hi. I wish we could get this guy fired. That would be so cool.
Okay, so finally understand the C-mix different from the B-mix. So that's good, because that's the B is viral, so that means that it's become a virus, the C is just cellular. Learned a new term called the proto-oncogene, just means that it is like the cellular, which can cause cancer, but it hasn't yet. Hasn't been mutated, yes, exactly. I'm studying my credit from C. We're gonna need to study that some more. But what causes that? That's where I'm lost. Is it that so like, because I thought the caspase caused the scrambylases and the phosphatidylserine.
So you get the cell death signal.
But who was sending this? The caspases? Because he said that the leaking mitochondria activated the caspases to make electrons and not what he's doing.
So it's like
that is what activates the scramblers, though that's what activates the caspase. It's kind of, I believe so. Being about it. But just moving on, I'll think about it.
also
I wonder if it's like I remember him telling me something about like it's like flashing and then it's like so maybe it's like slowly kind of like, and then once the mitochondria gets leaking, then it's just like green light. Maybe it'll be the right thing. It's kind of how I non-translocation or like taking things like the cis regulatory region is like it just got cut, so it no longer has the pliers part of it, so it just always activated. And then that is the actual coding region. So what I'm confused about is, do we just lose this part of the coding region? Like, what part of the gene is going in here? Is it just this little part of the coding region, or is it the cis regulatory region? Yeah, but like what part then is getting stuck in like what part is still like it would have to have its cis regulatory region, I guess. Yeah, because you said we're missing this, so this must be the left, like the left one for the transcription, I guess. Because you lose the pliers, so it's always turned off. So then that basically activates the non-kinase pathway. But then also more than just that, it also like stops the apoptosis signals, I guess, because it's no longer worried. But there's a drug for that, which is called the the drug, the antibody? There we go,
We'll do this for another minute.
You know what? I won't forget it now. Thank you.
Do 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 Rustam, what is one thing your group talked about that you thought was important today?
We kind of talked about CAR T cells.
Yeah.
Effects. We talked about immunotherapy.
Yep. Yeah. So these different therapeutic strategies, and so we're intentionally trying to show the different approaches and an example of each, just to get kind of appreciation of that's really different. So CAR T cell therapy is a really different strategy than giving someone a drug or some radiation or some surgery, right? It's a different approach like that. And then you, Sarah, what did your group talk about back there?
We talked about honestly a lot of stuff, but also the mutation in the transcription unit of RB, and how that reminded us of RAS, as it also had a mutation in the transcription unit.
Yep. Yeah, that's a very good point, right? Is that we saw earlier in our course, we emphasized really that mutations that cause RAS to be a trouble are in the coding region. And the mutations that cause MYC to be trouble are in the cis regulatory region. And so the mutation that causes ERB to be trouble is in the coding region, not just within the transcription unit, but very specifically in the coding region. And in that part, where we're going to make a shortened version, a truncated version. Yep. And Asira said you guys talked about a lot. So Lydia, did you talk about another thing?
We did. We talked about just trying to honestly like piece everything together that we've gone over the past couple weeks. We talked about how like the caspases relate to the scramblase, and then the scramblase itself is the flibase, which then triggers like the whole signaling apoptosis, and then just trying to get like those steps.
Yeah. Yeah, and I hope that you're starting to kind of appreciate that there's a certain style of topics in molecular and cellular biology, and you see like, oh, I get it. It's like a little kind of bedtime story thing where you're like, this is just a little thing. We should pay attention to this, and we'll talk about that. And then sometimes we go back to that same bedroom story, and we're like, well, actually, let's add that serum response factor to that, and just make it a little bigger, to kind of add it. Because that's really how I think, biologists go through their day, I think, is they learn a piece of something and they use that as a framework. I always use a Christmas tree as an example. They put up a Christmas tree and then they use that same tree and they start decorating it, with these other pieces. And biology is hard because you walk in and you're like, my gosh, that tree has like 400 ornaments on it. And you probably have a family history story for each one of those, Yeah, and I know a lot of people don't have Christmas trees, but that's just, a tree and you decorate it with stuff, right? You go out and just be adding your knowledge to that. You go to that place, right? And so, yeah, you can see we're kind of doing that, where especially we roll into this final chapter and we're like, we've been here. And we're like, yeah, but we're going to try to add a thing to it. Yep. Yeah. So we do have a couple more minutes. Let me show you 2 minutes. We have 2 minutes. So let me just show you this. Because in previous editions in this final chapter, our book did a good job walking through these features at the sort of cellular biological level of cancers. And we'll talk about those on Tuesday, but I'm mindful that we can't learn too much more on Tuesday because we have a test on Thursday. But we'll go through those, And so you don't need to scramble to write them down, but they'll be in our recording. You won't find them in our book, but just be aware that we want to learn that piece. And it's just a kind of a brief thing about what's this, what's that. It's not like a paragraph story. It's just a little thing about, well, that's weird. Like here's just a quick example of it. So the number 3. So if you take cancers out of a body and you put them in a dish with sterile and nutrients, those cancers can live forever. But if you take a normal cell out of her body and put it in a dish under the same conditions, those cells are mortal, just like the rest of our body. So there's features in the cell biology of cancer that are weird, distinctive, and that's one of them. So, there are features like that. So I just want to show you that. And then the other thing that we'll learn new is I mentioned that there are other viruses that contribute to cancer. And so I want to just show you this. So see this list? 4 most, now I shouldn't maybe say most common, but 4 really common ways that scientists began to learn about molecular mechanisms of cancer. Oncogenic retroviruses, jump down to chromosomal translocations, jump down to gene application. Those are all things that were around, as they were getting into like the 1970s, 80s. They're like, we're getting hold of something here. Here's this other one, and that is just totally different viruses that we find in nature. Don't cause cancer directly, but they can make an animal, a person or animal, susceptible to cancer. And the book list has a list of them. It's a very different story than oncogenic retroviruses. And the book just has a list. And in previous editions, the book did a good job going through some. And we're going to talk one on Tuesday. And it's this one. And it's HPV. And in the previous edition, I don't think that it's present anymore, it showed this human papillomavirus, so it causes genital warts, and it shows what some of the virus's gene products do. And look what they do. One of them inhibits retinoblastoma, a huge, famous tumor suppressor. And one of them destroys the guardian of the genome. Because the virus just wants to replicate. It wants to turn the cell into a DNA replication machine. And remember, retinoblastoma is inhibiting E2F from turning on the replication machine, activating hundreds of genes that are important for DNA synthesis. So it's not surprising to us at all. And we'll talk about that one example. Because it's like, oh my gosh. And there's a set of viruses that are like this. Is that a retrovirus? No. Is it an oncogenic virus? No. It doesn't cause cancer, but it's just getting its job done. Most people that get HPV, they heal, and they're fine. Some people kind of have a lingering infection, and that makes them very vulnerable to certain types of cervical cancers, like that. So we'll talk about that example. So we do have more to learn. That's good stuff. And, I'm trying to foreshadow a little bit. So, again, those are both things that really aren't in our textbook, so you don't have to sweat them. I'll try to package them in a way that we'll just move through them. But we'll also have time for review, and we'll be doing great. Okay? Awesome.